Thank you.
This is K. rileyi, and this is a gall laid by this parasitic wasp, which has co-opted the leaf tissue of its host for its larva.
So what'd you find?
Found K. rileyi right here on the red oak.
We found this specimen, which we identified by its distinct capello and gall, and many others after searching through the BU Nature Preserve and looking at the trees they've been known to be laid on. Do you need help?
I'll come down one way or another.
Okay.
Here's one.
However, we're not interested in this larva. Instead, we want to look deeper inside and see what kind of endophytic fungi may be inhabiting it and the tree.
We're picking a healthy leaf for comparison.
And for that, we'll need to take a closer look and head into lab to begin the lengthy process of isolation. I just finished disinfecting the immediate, thank you, the vicinity so we can then begin the isolation of the galls from the leaf. First, we disinfect. Then, we'll take the gall of the infected leaf. We'll dissect the gall into its different parts. Why are we cutting it in half?
To try to expose everything on the inside, and also allows us to disinfect it a little bit better.
The gall bits are then placed into the petri dish to hopefully grow.
There we go. Look at that. Perfect.
Alright, so now we're going to parafilm the petri dish. So the parafilm is going to do a couple of jobs. One, it's going to act as a layer of protection, and two, it's going to help hold the moisture inside of the petri dish so we can leave it for a couple days, see how the fungus grows, so we can actually properly then identify it later on. We then repeat this process for all collected galls, as well as the different healthy and unhealthy leaves we collected. Now that we've done all of this, the only step left is to wait and see what fungi grow.
Subculture fungi with us in the lab.
So we're going to first start off by once again decontaminating our whole space. These are the ones we are going to need to subculture. They can be noted because they have dual growth. Additionally, we're going to want to get rid of the bacterial growth to better isolate the actual fungi. So to do this, we take 2 new petri dishes. To disinfect the probe, we're going to stick that in the bacteriocinator. So I'm going to lift this up, slice that. All right, now that subculture is done, so we're going to want to parafilm that. But first, I'm going to take the other section and put it into a new one as well. And just like that, we have subcultured 2 of our growths, and we will see what they look like in a couple days. It's time to identify our fungi. If you remember, over the past couple weeks, we've been growing fungi found within K. rileyi wasp galls. And now, we're going to learn what species of fungi they are, using the polymerase chain reaction to amplify the DNA, as well as Sanger sequencing to read it. First, we'll prepare for PCR, meaning we'll need to extract a tiny portion from our fungi into a buffer to break the cell apart and free the DNA.
Oh, that's a sick shot.
With the DNA exposed, the solution is diluted, and the
Master mix.
is added to give the materials necessary for PCR.
Okay, now just repeat that.
40 times.
30 times, yeah.
This may sound complicated, but in reality, this process is mostly just moving liquid back and forth, and back and forth, and back and forth, and back and forth. The solution is lastly added to a thermocycler for the actual PCR process to occur, where in the region we want to amplify, in our case the ITS region as it varies a lot amongst fungi, making it ideal for identification, goes through denaturation, annealing, and extension. We then confirm PCR worked and our DNA fragments were actually amplified using gel electrophoresis, before lastly sending it off for Sanger sequencing.
We got both our chromatograms back for both the genes that we're going to be using to ID the fungi. And what we're going through is we have them both aligned, and now we're going to clean up our data.
We then blast this data into an online database to see which previously sequenced fungi it matches, leading us to finally identify our mystery species.
You may have remembered us looking for K. rileyi galls in the BU Nature Preserve a few weeks ago. Here's what we've been up to since then. As a recap, we wanted to examine the fungi in the gall to those on the leaf, and see how different species of fungi grow differently when exposed to oleic acid, a known fungostatic. We believed that fungi from the galls would grow faster in oleic acid, because galls contain oleic acid, suggesting the fungi would be more tolerant or resistant. To do this, we cut and isolated fungi from the capello, gall body, galled leaves, and non-galled leaves. Each part was placed in water agar to promote fungal growth. Then the youngest part of the fungi was subcultured. We chose to focus on 10 fungal species that were observed multiple times throughout our collection, or those unique to a specific part of the gall. These samples were also prepped for Sanger sequencing with PCR to determine their species using the ITS region of their DNA. Since this region is a highly variable, non-coding region, that makes it ideal for identifying fungi. To test the influence of oleic acid on each fungus, we subcultured each into 3 different agar plates that we made. PDA plates were our control, PDA plus tween 20, an oleic acid emulsifier, and PDA plus tween 20 plus oleic acid. We measured the diameter of each fungal growth every day.
As an example, this is our one with the tween 20, oleic acid, and PDA. So if you see on top, there's the fungi. On the bottom, I put a dot in the middle, and then using a ruler, I marked the 2 bisecting sides.
The daily average of the 2 measurements for each plate was used to calculate the average growth rate of the fungi, and was then put into our tables. We found that in general, fungi grew less in media containing oleic acid, and those originating from a leaf grew more each day than those from a gall. We also noticed that tween 20 also acted as a fungostatic, even without the oleic acid, affecting the fungi from the galls more than the fungi from the leaves. The results go against our hypothesis, as there was more growth in the fungi sourced from the leaves. This suggests that there may be something in the fungi that grows on the oak leaves that promotes their growth, or perhaps another fungostatic that is more potent than the oleic acid found in the K. rileyi galls. Ultimately, we hope that this will provide more insight into how insect galls and their fungal inhabitants interact with plants that they live on and are around. Endophytic fungi help plants withstand abiotic and biotic stress. For instance, they can help increase root production, nutrient acquisition, and production of secondary metabolites. However, fungi are also the #1 cause of disease in plants and can become pathogenic if the plant is stressed out. Learning more about what contributes to the growth of fungi may inform climate change mitigation in plant communities.